mechanistically, with respect to utrophin, we want to see the more uniform distribution, not only there where the repair takes place, but across the fiber where you usually see dystrophin, we now want to see utrophin taking over the function of dystrophin. So we also, with this, expect an increase in more mature fibers. With the MRI, we hope that we slow or stabilize the increase of fat, because we know from the natural history data that there is an increase in the fraction of fat in muscle tissue in DMD boys. And for the functional parameters, we are looking for a slowing or a decline, a slowing in decline or stabilization of these measures. The study, as I said, is ongoing. We're expecting first results by the second, third quarter, and this will then be followed by a placebo-controlled study, which should start somewhere in the last half of this year, subject to regulatory approval and discussions with the regulatory agencies, and hopefully this could be an accelerated approval program. And this then has to be supported by confirmatory trials. If you want to learn about our clinical trials, you either go to clinicaltrials.gov, or we have a website, utrophintrials.com. You can sign up there and get information about our clinical programs. Or you can contact Michelle Avery, who's here in the room with me, who's our patient advocacy person. That's her email and her telephone number. Thank you very much.
Grazie. Passiamo ora a Catabasis Pharmaceuticals. Joan Donovan.
Buongiorno a tutti, e mi fa molto piacere essere qui oggi. Allora, today I'm going to talk about itasolonexant, which is an NF-kappa B inhibitor that we are developing for patients with Duchenne, and we're developing this for patients regardless of mutation and potentially for all age groups as well. We are also a public company, and I will talk about our plans, so I need to include this. So you know that the lack of dystrophin is the fundamental issue in Duchenne. And the lack of dystrophin, along with mechanical stress, activates this protein called NF-kappa B, and that ultimately leads to muscle inflammation, muscle degeneration, fibrosis, and ultimately the loss of function in these muscles. We know that steroids have an effect suppressing inflammation, but they also have significant side effects. With itasolonexant, we've done phase 1 studies that showed no safety signals, and it was able to show that itasolonexant was able to target NF-kappa B. So currently, we are conducting the MOVE-DMD study to understand the effects of itasolonexant in young boys with Duchenne. Now, again, it's the absence of dystrophin with mechanical stress that activates NF-kappa B. And when this protein is activated, it has effects causing the destruction of muscle, preventing muscle from being regenerated, and this is shown in a cross-section of a leg of a boy, and it's shown by MRI, because we're using MRI as a key endpoint in the study. Additionally, activation of NF-kappa B causes inflammation and fibrosis, and it also prevents stem cell replication and stem cell regeneration of muscle. So we've shown effects on all three of these in preclinical studies and animal studies, in the lab of Professor Lee Sweeney. So we've designed the MOVE-DMD study in 3 parts, so it's a bit complicated, and I'll go through that. We have enrolled in this study boys aged 4 to 7 who are not yet on steroids, and that's because of the potential overlap in mechanism between steroids and etosolenexant. In the first part of the study, which I spoke about here last year, we looked for 7 days. We gave the boys 7 days of etosolenexant at 3 doses, and we looked at safety. We looked at whether or not there was effects on NF-kappa B, and we also looked at the absorption of the drug in the bloodstream. What I'm going to talk to you today is the results of this part, Part B, which was a 12-week randomized, double-blind, placebo-controlled study with 2 doses of etosolenexant. We actually took the higher 2 doses because they were safe and well tolerated in the first part. And we also included a group of placebo patients. After the end of 12 weeks, all of the patients then started on etosolenexant at the 2 higher doses, and they're continuing for 36 weeks. What we did to assess the effects of etosolenexant in the study was to look at the beginning of Part A, at the beginning of Part B, at MRI, and it's a particular marker of MRI, T2, which shows the effect, or we thought would show the effect of inflammation in muscle, as well as the fat fraction. We also looked at the timed function tests that are appropriate to this age group. So we looked at 10-meter walk-run, 4-step climb, time to stand. We did not use the 6-minute walk test because that's not very helpful in boys as young as age 4. And we've continued then to look at these tests periodically through Part B and also through the 36-week open-label extension. So in the first part of the study, one of the important things was etosolenexant is given as capsules, and these are a Tic Tac and an M&M, so these are relatively small. And it was very good to see that even the 4-year-old boys were able to take these capsules quite readily, and they did good with them. They even went up to the larger size capsule during the study. And in this first part of the study, etosolenexant was well tolerated, and the most common adverse events were soft stool or diarrhea, and it didn't prevent any of the patients from going on to the second part of the study. Now, we looked at MRI, and this is the T2 relaxation time, which basically looks at how water is mobile within the muscle, and that's what we're looking at here with the T2. We chose this because the imaging DMD group, who we've been working with, have shown that steroids had an immediate effect on T2. And what we hoped was, if we did see an immediate effect, then that could make us move much faster into a registration study, into phase 3 study. But we also included the time function test, the North Star ambulatory assessment, muscle strength, parent impressions, the PODCI, as well as the fat fraction. We knew that all of these would be changed over a longer period than 12 weeks, and we were planning to look at those predominantly in the extension study for information for future studies. Now, when we enroll patients in a study, you want to make sure that the groups are as balanced as possible. So we allocated boys based on their age to try to make it even, and also on how quickly they walked at the beginning of the study. And we saw that while the age was very similar across the groups, there were differences in that the boys in the higher dose of etosolenexant were diagnosed earlier than boys in the placebo group, and they had their onset of symptoms earlier, more than a year and a half earlier. And that was reflected in the 4-step climb and the time to stand, that these were on average longer than the boys in the placebo group. So it ended up that the groups were a little bit different. And this is 31 boys overall with 10 in each group. Now, for the primary endpoint at 12 weeks, we were looking at MRI. And we knew that this would increase in the placebo group, which it did over the 12 weeks. While we saw less of an increase in the 100 milligrams per kilogram group, we had anticipated seeing more of a decrease there. So while there was a difference, it did not meet the primary endpoint. And what we've trying to understand and working with the experts is to understand whether the effect of steroids on the water in the muscle may actually be a separate effect from the effect on NF-kappa B and inflammation that we see consistently in the preclinical studies and also in the biomarkers that we have looked at both in the boys as well as in adults. So we also looked at the time function tests. And in the 8 months between the Part A and Part B, we saw that all of the boys had decreases in the speed of these tests. So we're looking at speed here. So up is what we want to see compared to the placebo. So we saw that the 10-meter walk-run and the time to stand were decreased in the placebo group and less so in the 2 treatment groups. And in particular, the time to stand, this is not changed. It was slightly decreased in the 67 milligrams per kilogram group, the lower dose group. But in the higher dose group, despite those boys being at a somewhat more advanced stage, they were stable. We also looked at the 4-stair climb, which others have used as their primary endpoint in a registration study. And we saw that there was an improvement, particularly in the higher dose group, in that they were faster versus baseline. Now, none of these are powered for statistical significance. We didn't expect to see changes at this point, and certainly not statistically significant changes. The muscle that you use most for climbing stairs is the quadriceps muscle, and one of the components that we look at is the vastus lateralis. And that was, instead of having an increase in the placebo, what we wanted to see is that it was flat or decreased, which we did see with the vastus lateralis. We also looked at the North Star, which is a composite of 17 different tests of function. And as would be expected in the placebo group, there was some decrease. Typical is several points per year. And it was stable in the etosolenexant. So there seemed to be consistent improvement or stability in the 100 group versus placebo. And we are planning on focusing on that group and looking at those more as we move into the open label extension. Now, very important in a phase 2 study is safety. We did not see any safety signals. All of the boys completed the 12 weeks of the study, taking the full dose of the medication. The most common adverse events were GI, mild diarrhea, occasional vomiting, and this was in the context of about 8 patient years of exposure in the 4 to 7-year-old boys. So to summarize, if we had seen a change in the primary endpoint, we would have, this would have paved the path to more rapidly moving into a phase 3 study with a shorter, so to be able to do a shorter trial without muscle biopsies. Unfortunately, we didn't see that early signal that we were hoping for, but we do see that the etosolenexant, particularly the 100 milligrams per kilogram group, the higher dose group, consistently showed numerical improvement versus placebo, even at this relatively short duration of 12 weeks. And as anticipated with this size of patient group, these changes weren't statistically significant. We didn't see safety signals, so we're very comfortable with the dose of 100 milligrams per kilogram. And we only presented these results publicly, released them a couple of weeks ago, so we're still very much looking through and understanding the effects of itasolonexant in this group. But also, very importantly, the boys are continuing on for almost a full year of treatment in this part of the study, and we'll have a lot more to share in the coming months about the effects and what we see at the 6-month point and beyond. And lastly, grazie mille to the patients, to the families. We had support from PPMD and MDA for travel, and we had support for patient groups in the U.S. also for reaching out and helping recruit, helping make the parents know about this study. So, grazie.
Bene, siamo perfettamente nei tempi, per cui grazie. Ci spostiamo sempre su meccanismi generali, quindi Eric Hoffman, Vamorolone.
Good morning. Thanks for being here. So I'll speak about the vamorolone program this morning. I should just point out that the crystal structure, so that's what the drug looks like. That was recently solved, and it looks like glucocorticoids as we expect. Just a quick disclaimer. So this is under development for DMD in clinical trials. More complete data is published, available online or by request, and this is an investigational drug and has not shown proven benefit. Long-term safety has not yet been clearly demonstrated. If you have any questions, there's an email here and also at the end for contact information. Also disclosures, so I'm involved in 3 spin-off companies from academics, but they're all under a venture philanthropy model, which I'll come back to and talk a bit about that later. But I also work at a school of pharmacy in New York, but here I'm representing Revirogen Biopharma. So the goal of this program was to try to understand why glucocorticoids, prednisone and deflazacort, why they do improve strength in Duchenne, but also why they have all these side effects, and to try to separate those and create a new drug where we would keep and improve aspects of efficacy or clinical benefit, then remove aspects of safety concerns or side effects. In understanding what Duchenne muscular dystrophy is, what's wrong with the muscle, what dystrophin does, we also try to target different parts of that disease process and try to add new parts to the drug. So this is what's called a dissociative steroid. Now, a very important thing to point out is vamorolone is not a glucocorticoid. So it is not in the same class of drugs as prednisone or deflazacort. It's a first-in-class, a different structure, and has different functions that I'll go over in the subsequent slides. So we did a whole series of mouse studies, as you've seen in many presentations. And the key thing is that we could clearly see we were losing some of the side effects, such as the growth stunting. With prednisone, you see the same growth stunting as you do in boys with Duchenne muscular dystrophy with both deflazacort and prednisone. And even if we went to high doses with vamorolone, we didn't see that. So we didn't see the bone changes or growth stunting. So I'm not going to go over a lot of the data because most is published. But in this slide, I'll spend a bit of time because this summarizes everything we know, not only from studies in mice, but then studies in humans, because we have finished the studies in about 85 adult volunteers, and we're currently in boys with Duchenne muscular dystrophy. So most of my presentation, I'll go over the trials that we are ongoing and have planned, and I'm not showing you much data, but I'll just summarize it here. So as I said, it's a dissociative steroid, which means you're taking different parts of steroid activities and separating them. You're increasing some, decreasing others, adding some new parts. Because there's many activities, I've separated it all into different categories here. And clearly there's 2 parts. There's efficacy, or benefit, and safety, which is side effects. And so those is one way to dissociate those 2 different topics. So in terms of efficacy and benefit, Anything that's black is similar to glucocorticoids. Anything that's red has become different than prednisone and deflazacort. So under efficacy, we've retained the NF-kappa B inhibition that Dr. Donovan just mentioned in the last presentation. Because, as I'll show you in a minute, we've removed most or all side effects, we can increase the dose much higher than prednisone or deflazacort. So in adult volunteers, we've gone to 30 times prednisone dose and still saw a lack of side effects. So we could have kept on going, but we just gave up. We ran out of money. So we've gone to 30-fold, and that's what's called our NOAEL. Now, one of the things we've added new is a membrane stabilizer. So this we've shown in vitro. This hasn't been shown in humans yet, but we're stabilizing the plasma membrane, whereas prednisone destabilizes it. So we're protecting against dystrophin deficiency. Another drug that's become clear is beneficial to Duchenne heart is eplerenone. Eplerenone is what's called a mineralocorticoid antagonist. It's used in heart failure frequently, and there's nice publications in controlled studies that this aids Duchenne muscular dystrophy heart. So it helps benefit or shows efficacy in heart. Well, vamorolone we've changed into eplerenone. So it has very similar activities to eplerenone, same potency, same effect in heart and mice. So and it also helps the MDX heart problem. Prednisone and deflazacort don't do that. So they are an agonist of MR, not an antagonist. They actually have the opposite activity on heart as eplerenone. So we've turned a side effect of heart of prednisone into a benefit through this change in the chemical structure to an antagonist, and that's new. We also know that we synchronize regeneration. There's publications on that, but also prednisone and deflazacort do that. Now, that's all efficacy. So just to summarize, we've retained the anti-inflammatory. We've added membrane stabilization. We've added an antagonist, a mineralocorticoid like eplerenone, and we synchronize regeneration. Now, for safety, we've studied all these different aspects of safety concerns of glucocorticoids. And all the data that I show here is very consistent both in mice and in our human studies to date, or adult volunteers. In adult volunteers, it's difficult to study efficacy because your adult volunteers don't have Duchenne. They're healthy. They are healthy. So we can't look at efficacy, but we can look at safety in adult volunteers, mostly because we have what's called biomarkers. For many aspects of the safety concerns of prednisone and deflazacort, we can just look at a drop of blood and clearly see these safety concerns and if they're happening in an adult volunteer. So one thing we've seen consistent with mice is this loss of stunting of growth. We measure this by bone biomarkers in the blood, osteocalcin, CTX1, and we see even at very high dose, we're not changing any of these biomarkers, and we're likely not stunting growth. Loss of delay of puberty because of adrenal suppression. In adrenal suppression, we see about 1/100th, and there's a concern of adrenal crisis because we're not suppressing the adrenals in the doses we're using for Duchenne. We've likely lost that risk of adrenal crisis. All the metabolic disturbance, which causes the appetite, the weight gain, everything says we've lost all of those, again, both in mice and in humans by biomarker studies. We've lost this MR agonist activity, and we've lost immune suppression. So as far as we can tell, everything in mice and humans is consistent so far, but obviously for efficacy in Duchenne, we need to do the Duchenne trials. Now, a quick note on the business model here, because it's much different than most you've heard about. We've worked with governments and foundations internationally to de-risk this program, which means that many of the things we've done throughout the drug development program were independently validated by the government. So the government would hire another group, it's called NIH Trend, and they said, you test this and see if you come up with the same answer as Revirogen. And so that worked out very well. And they also aided in the next steps of drug production and formulation. Treat NMD TAC group reviewed the program and helped de-risk. And to date, we've raised $35 million in what's called non-dilutive capital, again, through foundations and governments, and also a recent agreement on an option deal with Actelion Pharma in Switzerland. Part of this is of the major grants given to us by foundations, there's a return on investment of those grants. So there's a 400% or 450% return to the foundations based on later drug sales. And as a result, this program is run by an international collaborative of academic key opinion leaders in Newcastle, in the US, and worldwide. And it's really focused on innovations in clinical development for Duchenne. So briefly, again, I won't go over any data, but the phase 1 was in about 85 normal volunteers. That was completed in 2015 with support from the Muscular Dystrophy Association, joining JAC, Duchenne Research Fund, and Duchenne Children's Trust, and we're very grateful for that support. We couldn't have done it without it. The DMD program, we're currently in phase 2A. That's supported by the National Institutes of Health, both NIAMS and NINDS. And the 2B program will begin this summer, and that's supported by the European Community, the Horizons 2020 program. We have a focus on innovations, data-driven clinical endpoints via natural history studies, data-driven pharmacodynamic biomarkers. We've done parallel EMA and FDA guidance, which is also unusual. And as I mentioned, we have international key opinion leaders to run the program, Kate Bushby and Michaela Guglieri in Newcastle, and Paula Clemens in the University of Pittsburgh. So now the last slides, I'll just describe the clinical trials in Duchenne. The 2A and 2A extension started in July last year. We're enrolling 48 DMD boys, ages 4 to 7, that must be steroid naive. We go over a very large dose range. Both EMA and FDA have said you need to really do good dose finding studies. Do a broad dose range and find that safety efficacy window. So we're currently finished the 0.25 group. This month we're going to the 0.75 group, which is similar to prednisone. And then we're going to 3 times prednisone and 10 times prednisone. We have 12 recruitment sites, US, Canada, Sweden, UK, Israel, and Australia. We expect to complete enrollment around April or May. The design is a 2A, which is a 2-week treatment, then a 2A extension with a 6 months treatment. All patients are vamorolone treated, there is no placebo, and then there's the option to enroll into a 2-year extension program. The first DMD patients have enrolled into the 2-year extension. So what is involved in this phase 2A trials? There are no muscle biopsies. We've designed the trials with parent and family involvement to try to reduce the burden on families. And the foundations helped us pick the endpoints. We had many endpoints that were similarly powered, but we ended up using time to stand because the parents felt that that was most relevant to their quality of life. Dosing of the drug is done at home once each morning. It's just a liquid suspension. We easily adjust the dose. We reimburse all travel and stay, and so far we've been able to give reimbursement checks within 2 weeks back to the families, and there are no overnight stays in clinic, so it's all outpatient. All patients are treated with vamorolone. There is no placebo, no prednisone or deflazacort. The phase 2A visits happen over a time of about 6 weeks. There's a screening visit, a 2-day visit, and 4 weekly visits. And then the phase 2A extension is over 6 months with 2 2-week visits, 2 weeks apart between the visits, and then 5 monthly visits. And then the long-term extension is visits every 3 months. The exclusion criteria is previous treatment with traditional steroids such as prednisone or deflazacort, and must be enrolled prior to 7th birthday. I know that parents and families are concerned about failing screening. So if you go to a trial and then you get screened, will you fail the screening and not be able to enter the trial? We've only had 2 failures to date. One was lack of sufficient immunization for chickenpox, but the child was just re-immunized and then entered into the trial later. And 1 in 12 children was unable to stand from the floor, even at this young age range. And since that's our primary outcome measure, that child unfortunately was excluded. As I mentioned, we have all sites active by the end of this month. We'll reimburse for travel and stay to the closest site, and I think I went over all this. Okay, and these are the 2A sites that I've mentioned, and I won't go over them. Okay, the last slide's on the phase 2B. This should start this summer. There's 100 DMD boys, same age to 4 to 7 years. The study director is Michaela Guglieri. CRO is Trins LLC. Sites are both Synergy and non-Synergy, and I mentioned the Horizons 2020 funding. There'll be 30 recruitment sites in 10 countries, and an emphasis on Europe because of the Horizons 2020 EU funding. It is blinded and randomized with a 6-month treatment period in the 2B, and then a 6-month extension study, all of them vamorolone, and then the option to enroll in the long-term extension. What is involved in these? Well, the initial trial that's blinded is 6 months, but there is a placebo and there is a prednisone arm. So there's a 50% chance of vamorolone, 1 of 2 doses, 25% placebo and 25% prednisone. But once that 6 months is done, everybody can receive vamorolone if they like. So we stop the placebo and prednisone. All patients will take both a pill and a liquid medicine each morning, and again must be steroid naive and enrolled before the 7th birthday. No muscle biopsies, 8 visits over 6 months, and RevereGen reimburses for all travel and stay. And these are the sites at this point. These have all now, so we're starting contracting with these sites. We'll have 3 different groups of sites. US and Canada will probably initiate first, then the UK, Sweden, Israel, and Australia, and then all the other countries as a next tranche. So if this works, and I think we'll have in the next few months how well this is working or not in this unblinded study supported by biomarkers, where will we go from then? So we have a series of clinical trials both in younger boys and older boys, boys that are even on steroids or off steroids. So these are all mapped out with the EMA, what's called a PIP, Pediatric Investigation Plan. And those would be, as we learn more and know which dose we're using and have proven safety and efficacy, to expand those clinical trials. Just the last 2 slides, RevereGen functions under a different business model, as I mentioned, a program designed and run by academic key opinion leaders and stakeholders, parents, foundations, governments, extensive de-risking. Many aspects of the program have been independently validated. Everything's published. We have 9 publications to date. And a key issue is we're not risk averse, because I know that as you as families, you want everyone to take risks to try to move the needle, move the field forward as fast as possible. In our position from academic and partnering with foundations, we are not risk averse. We are very willing to take those risks, in part because we're not answering to stockholders. We don't have to worry about publicly traded stock and having those stockholders say, well, you can't take risks because we need our return on investment. So I think that's really important to us and to this program because of outcome measures, working with regulators, really trying to push things very aggressively forward. And lastly, we have innovations in clinical outcome measures. We're using time to stand from floor as our primary clinical outcome measure. Extensive secondary outcomes, including MRI in the 2B, registration focused on younger age of patients with a subsequent extension of labeling, and we're very easy to design our trials because we're benchmarking against steroids. We have to beat steroids, prednisone and deflazacort. You've heard through this conference a number of times discussion of biomarkers, and biomarkers are very important. The US just passed legislation 2 months ago that really wants lots of drug development to focus on biomarkers. Now, what does that mean? What it means is from, say, a drop of blood, you can see what the drug is doing, both in terms of efficacy and safety. That's the goal of biomarkers, so that the changes in the blood, perhaps very quick changes, maybe just hours or days or week after drug, that you can predict what that drug's going to do to the patient's function, both with safety and with efficacy. So we've used very innovative biomarkers throughout this program, both for safety and efficacy. It's been very interesting discussions with both EMA and FDA. So I'm just outlined here how we're using them. We have for safety, secondary safety outcomes. We have markers for adrenal suppression, insulin resistance, bone turnover. In the 2A and 2B, we have exploratory safety outcomes. We think we've found the first biomarker for stunting of growth, for example, where after just a week of drug of prednisone and deflazacort, we can see that that drug is going to stunt the growth 2 years later. That's what the value of a biomarker, acute and objective readouts of drug effect. Metabolism, kidney side effects, and then exploratory efficacy outcomes in terms of pro-inflammatory proteins. We're measuring these just hours after initial treatment. So in these trials, the very first dose of vamorolone, 5 hours later we take a blood sample from each child and we measure these biomarkers. And we know they're accurate. All these in the secondary safety, we already know we can see metabolic disturbance, we can see adrenal suppression just 5 hours after the very first dose of drug. That's the goal of biomarkers, to make it more objective, to more acute, and to speed up the drug development program. And here's an example of newly discovered biomarkers that we did together, Yetri Pathout and others, the Synergy Group, Craig McDonald, and these are published. But it's just to show a biomarker that in normal kids, in blood, you'll see like this lymphotoxin at the top is low. When you have a kid with Duchenne, it goes very high. And when you treat the child with Duchenne with prednisone, it goes way down again. And we know each of these proteins is pro-inflammatory. They're working towards making the muscle sick through pro-inflammatory pathways. We know this is one of the mechanisms of action of deflazacort and prednisone, and this is what we want to keep through this, and this is what we're measuring both hours and weeks and months after drug treatment. So in just acknowledgments made possible by National Institutes of Health, U.S. Department of Defense, EU Horizons 2020, and many foundations that have been very gracious in helping support this program. And these are the symbols of these different foundations. Thank you.
Thank you. So, adesso Michael Biggs, Pfizer. We move on.
Thanks very much, Valeria. We're getting the slide. And thank you to the organizers of this wonderful conference for the invitation to talk to you today. And I'm going to talk to you about domagrozumab, which is a monoclonal antibody to myostatin that's currently in phase 2 pivotal clinical trials. I am an employee of Pfizer and need to show a disclaimer about the statements I'm going to make. So we believe this mechanism, and you heard about it from Leslie Jacobson yesterday, presenting on the BMS drug that acts in a similar way. We believe that there's potential to delay disease progression in Duchenne, regardless of genotype. The study I'm going to talk about is B516-1002, and it's ongoing in multiple countries around the world, but including in Italy, and there are 4 sites open or nearly open in this country. And I just wanted to mention at the end of the talk some investments that Pfizer and the rare disease group at Pfizer have made towards AAV gene therapy. So myostatin is a small protein hormone that's produced by muscle and acts on muscle that is well characterized to inhibit the growth of muscle. It was initially described in the 1990s, and very rapidly Catherine Wagner did some experiments to knock out myostatin in the MDX model, and showed that although there wasn't a dramatic improvement in the overall morphology, there was certainly a reduction in the degree of fibrosis and fatty replacement in the MDX muscle in those double knockout animals. Subsequently, experiments were conducted in academia to demonstrate the intervention using a therapeutic drug to have the in the MDX model, and similarly relative to the wild type MDX, to the MDX model, those treated animals seem to have much reduced fatty replacement and fibrosis. This initiated a number of investigations to see whether inhibiting this mechanism could help out in Duchenne. So we developed an antibody that we felt was suitably potent and specific to progress another experiment in this therapy area. And in preclinical studies, we were clearly able to show an increase in lean body mass in mice, an increase in specific muscle weight in the MDX model as well as in normal mice, and a corresponding increase in grip strength measures in the MDX model. In larger animals, our drug was able to increase the lean body mass in a dose-dependent manner, and as measured by DEXA and by MRI, the thigh volume by MRI, we were able to show after a few weeks of dosing a sustained effect on the lean body mass in monkeys. Now you'll be aware, you've seen this slide many times, of the progression as seen by MRI in the natural history of Duchenne from relatively normal-looking muscle to muscle over many years that is largely replaced by fat and fibrotic tissue. And you'll equally be aware of this data from imaging DMD again that seems to suggest that there is at least a correlation between the level, the proportion of fat in a particular muscle and a functional consequence here as demonstrated by the 6-minute walk. So as the fat content gets sufficiently high, function seems to decline. Whether this is a correlation or is causal in any way, it remains to be seen. So we were very encouraged in our phase 1 study with domagrozumab when we dosed healthy adult individuals just 3 times over a month. This is a dose that corresponds to the medium dose in our Duchenne study, that over a period of 4 months we saw a sustained increase in the ratio of muscle to fat, which suggests that it moves muscle metabolism at least in the right kind of direction that may have some benefit in Duchenne musculature. So this is the study that's currently running. It's quite a complex study, but it is a randomized placebo-controlled study. You'll see this placebo group here for a full year. Boys entering the study have 2-to-1 odds of going on to active drug, and then they dose escalate from a low to a medium to a high dose. Initially, at each of these changes, there was a very careful safety evaluation before the next dose was allowed. We've recently heard from our data monitoring committee that actually we don't need to do that anymore. They're sufficiently confident with the safety of this regime that we're just dose escalating without that particular individual safety review. So that's been very good news. The other change that's happened recently to this protocol is that we were initially selecting boys in the age group of 6 to 10 years. We had sufficient data last year from natural history studies to believe that we could increase the age range without compromising the statistical underpinnings of the study. So we've done that, and that's obviously opened the study up to a larger proportion of boys. The boys need to be walking on a stable dose of steroids, and they receive in this study a monthly 2-hour infusion. And then evaluations for functional, for function every 2 months, and with MRIs every 4 months. So there's a lot going on in this study, and we're going to have a very rich data set at the end of it. Our primary endpoint for this study is the 4-stair climb, selected based on partly the mechanism of action of our drug, but also the excellent statistical performance characteristics of this measure. But in addition to that, we are measuring some of the other functional endpoints and other imaging and biomarkers that I won't go into at this point. Now, to ensure that all the boys entering the study receive the active drug at some point, the second year of the study, those boys who have been on placebo will then receive the dose-escalating course of the drug. Those initially randomized to active drug, some will stay on the high dose and some will be randomized to placebo. And this group generates information for us on how long the effects persist, and also it's an opportunity for us to understand the immune response to our drug and whether there is one that is of concern. We have an open label extension study that is currently running, and all boys completing the study have the option to enroll into that, which will continue to collect both functional and safety data over a period up until either the program is terminated or it's approved and marketed. The whole study is overseen by an external data monitoring committee that reviews data ad hoc, but certainly every 4 months, and constitutes some Duchenne experts as well as a cardiologist and other experts on the committee. So what's the situation with this study? We are now, we have 90 boys enrolled, 40 boys have been in the study for over 1 year. 3 boys have completed the study and entered the open label extension. And from a safety perspective, things have gone very well. I think the IV infusions are well tolerated. We have no emergent safety events of concern. We did have recently a serious adverse event. Now that is there's a technical definition for that, which means anything that occurs which is serious enough to require admission to hospital. And this particular boy had appendicitis and is now recovering well post-surgery. So we don't believe that has anything to do with the treatment in this study. And we'll also say the Data Monitoring Committee have recently given us permission to reduce some of the safety monitoring in the study, which does simplify it to some extent. We have a study website which can provide you with up-to-date information and requirements for entering the study and details about what happens in it. And that's publicly available at that website. So in 2 minutes, I just want to highlight to you that Pfizer have made some regard as quite risky, but certainly an adventurous and innovative investment into AAV research. The field has clearly moved on in the last 20 years to a great degree. There's much more confidence in the safety of adeno-associated virus gene therapy and its ability to deliver effectively. So we have partnered with Spark Therapeutics in our first program focused on hemophilia B, and that program has gone very well, producing some data recently to suggest that there was very good transduction of factor IX, so the activity is at a high level, and these individuals are able to stop their twice-weekly factor prophylaxis while they have levels at this in this range. And we're continuing to observe this first cohort and planning for a phase 3 study in the not-too-distant future. So boosted really by the success of this program, we were very fortunate to have good relationships and interaction with Jude Samulski, and the company acquired his company, Bamboo Therapeutics, in August of last year. And it probably hasn't escaped your notice that one of the programs that Jude was progressing very aggressively and has been working on for a long time is a Duchenne gene therapy program. So we are hoping to move this molecule into the clinic in the not too distant future. In fact, there are a couple of individuals from Bamboo at this meeting, Sharon Hesterly and Mary Smith. So do identify them and grab them if you have any questions about this program. But it's a very exciting development, and we're really looking forward to seeing how this progresses. So I think I'm just at time, so I'll stop there. Thank you.
Possiamo avere qui I relatori per domande? Can all the speakers come to the table, please, so that we can get questions from the audience? Thank you. Direi che abbiamo sentito varie possibilità, indipendentemente dalle mutazioni, quindi su meccanismi più generali, per cui cogliete l'occasione per chiedere come magari vorrete approcciare la presentazione di nuove possibilità di entrata nei trial. Inizio io. So, I'll ask you in English, and then I will give it to. So, for the edasalonexant, the drug will be not against steroids, just placebo. Quindi nel trial presentato con l'edasalonexant il farmaco sarà verso placebo, e perché forse l'ho perso, ma non mi è sembrato di sentire che è contemplato lo steroide come ramo di confronto.
Yes, in this study, we are comparing versus placebo. There is a great deal of information on the effects of prednisone and deflazacort. So what we are comparing to in the longer range part of the study is the information that Imaging DMD has on MRI fat fraction collected over a longer period of time with the same investigators in the same site. And we're also going to be able to compare then to their data on time functional tests, again, by the same investigators, the same physical therapists. So we think that it is important to compare versus placebo. That is our plan in the United States, and we will continue to get input from the regulatory authorities as well.
There's a question down there.
Sì, una domanda per il professor Hoffman. Volevo sapere, perché forse ho perso un passaggio, volevo sapere se per la fase 2B del vamorolone sono inclusi anche dei centri di reclutamento in Italia, perché ho visto che per la fase 2A non se ne parla.
So the phase 2B sites I listed on the slide. I thought that Italy was included there, but I don't remember.
2A.
2A. The 2A is limited to synergy sites. So the Synergy network was a network we established some time ago, and so because we have some muscle function tests that require the training of a Synergy site, so there were there was one Synergy site in Italy, but we decided just to go with those couple countries. So there is no option for the 2A at this point because it's too late, but for the 2B we anticipate enrolling in Italy.
Perfetto, grazie.
Salve, sempre una domanda sul vamorolone. Ho visto con piacere insomma, l'aver minimizzato tutti gli effetti collaterali più importanti dei vari cortisoni e sarei curioso di sapere se avete fatto anche un controllo sugli effetti comportamentali che solitamente il cortisone dà negli individui, tipo molto nervosismo e problemi di questo genere. Grazie.
So we're certainly measuring behavior in the studies. With mice, it's challenging to look at behavioral changes, so we didn't measure it there. In the phase 1 data in the 85 adult volunteers, we did not observe any behavior that seemed related to drug. But in all the Duchenne trials ongoing, we are looking at that. We don't know yet. Thank you.
Altre domande?
Grazie. Una domanda riguardo Edasalon Accent. Visto che non è stato raggiunto l'endpoint primario con la risonanza magnetica nelle prime 12 settimane, però ci sono comunque dei parametri funzionali che hanno mostrato comunque dei miglioramenti a uno dei dosaggi. State valutando l'ipotesi di modificare lo studio prendendo altri endpoint primario, cioè cambiando l'endpoint primario dello studio o comunque di iniziarne un altro dove ci sia un endpoint primario su cui ci sono stati dei dati più favorevoli?
So the important thing in phase 2 study like this is our first study, proof of concept. And what we want to get from that study is information on how to design a larger phase 3 study. And for that, we need the information on the endpoints. What's important for a phase 3 study from a regulatory point of view are the functional endpoints that we are measuring. We're also measuring the fat fraction long term because, as many people have spoken about, that's very important, very correlated with functional measures. So we are anticipating, these are our pre-planned analyses to look at these secondary endpoints. And as far as going to a registration study, the important thing is the information we get from them. It's not necessarily what they're called. For a phase 3 study, you have to meet your primary endpoint, but for a phase 2 study, it's for our information to share and what's the basis for moving on to phase 3. Okay? Grazie.
Bene, allora i relatori saranno ancora con noi. Se abbiamo altre domande gliele chiediamo mentre ci prendiamo il caffè, lo portiamo al tavolo così recuperiamo il tempo. Okay.